So how to get or how to create this structured file for genomics analysis? You can take the raw FASTQ files and use the Taubot bioinfo platform to set up an example pipeline, or set up a pipeline that is going to convert these FASTQ files into a table of VCF files, structured data, that we can employ for visualization or further downstream analysis. Let's go to let's cut here and go to a demo pipeline, or set up a demo pipeline in the Taubot bioinfo platform, so that we can get a taste of how to set up a genomics pipeline or genomics data pipeline in the Taubot bioinfo platform. Those who have credentials, please log into server T bioinfo, and when you log in, you're going to get a screen like this. And in this screen, please follow along as you have. And if you are if you have any hiccups, or if you feel any necessity to ask me to wait or proceed, we can do that and have an interactive session. So please log into server T bioinfo platform, and under mutation variant and parallel analysis of mutation variant data. So you generally have we generally have the collection of different pipelines, or setting up different pipelines and algorithms for different types of analysis, as Elia had explained it to you. So for several of these analyses, we have prepared what we call as demo pipeline, where users, you can get an experience of setting up an analysis pipeline, and understand how each and every step that is involved in setting up this pipeline is being incorporated into this analysis into the platform. And you also get access to the processed or structured data at the end of setting up this demo pipeline, which you can download and perform your analysis, downstream analysis, or practice your analysis that you have been learning in the Learnomics platform. So let's go here in the mutation variant analysis and the mutation variant data section. Please click this down arrow, and then you have several demo pipeline that has been carefully constructed or carefully designed for you. So let's check one of those. So you're going to get experience of setting up a pipeline from several different options. So in the demo pipeline, and also when you are going to set up your own pipeline, you get suggestions for your next possible steps that you can do, or that you can perform within the available data, depending upon what you have selected for the previous step. So since this is a demo pipeline, the necessary files have already been uploaded and they have been referenced. So we start it, and we start and say okay. And before selecting each and every option, you will get a short information window of what that algorithm is going to perform. For example, this algorithm is going to clean up your PCR amplification duplicates and amplification biases, and you click OK, and then you have several of these from several of these options. Now we have only one option available for our next logical step. And yes, Omnia, please. Yeah, please share your question with us. You can share it in the chat box also. I think you're still muted. That's okay. So yeah, so to go forward, let's click next step in the pipeline, where we are going to, after cleaning up the PCR amplification artifacts, we are going to map these sequences or map these short reads onto the reference genome. And then we click OK. And then our next possible output is our next possible option for this pipeline or for this setup is highlighted as differential variant calling algorithm. So you click OK, and then you use Somatic Sniper to compare the variants that are calculated from two different biological types then you end the pipeline here. So once you end the pipeline, you get access to yeah, Iman, though in the same demo, you have more options. I have more options than your screen. When you click some of those options, and if you follow either of those options, you are going to get access to a different demo pipeline. One of them is going to be a visualization pipeline. The other is going to give you output like like this, a tabular output like pipeline. So depending upon what you have clicked so far, you will get different options, and you'll get you just try to click through them and understand what is what is being analyzed through the output files. Is there a link of recording in this demo? Yes, there are. Yeah, there are recordings that talks about these demo pipeline, and this Sonalika will share with you where to find those recordings. And for the purpose of today's session, let's move forward. So this is how you can perform a genomics analysis and then download the variant calling file. Since this file is too big for current session to complete download, let's move on to the PowerPoint presentation. Okay, can you see the screen? I think yes. So a similar omics data, another example for an omics data is transcriptomics data. So such data, transcriptomics data, represents the mRNA expression, which is one of the very good way to analyze what is actually going on inside the cell by observing active transcription of protein coding genes. And when we normally know that DNA is first transcribed to RNA, and then RNA is translated to protein, and depending upon the conditions and the specific conditions of the cell and the stress that cell is undergoing, these expression of the protein will change and will be characteristics to the phenotypical condition of the cell or the sample that we are analyzing. So to compare between genomics and transcriptomics analysis, transcriptomics analysis is simply not a subset of genomics analysis by leaving out non-coding part. Protein coding regions generally in the DNA or in the genome contain large stretches of non-coding sequences called introns, and when these introns are spliced out in RNA transcripts, then we will have different splice forms for the same gene being encoded or the same gene being developed. And depending upon the condition, depending upon the phenotype of the cell, some of these splice forms will be expressed more often than the other. And such an analysis is going to help us understand the state or help us understand the biological condition of the cell, or will be extremely useful in performing a differential gene expression analysis, which is just going to compare the expression levels of cells, for example, of tumor cells and normal cells. So how to set up such a pipeline? We can again go to the demo pipeline in the Tabot Bioinfo platform, and we will now, for this analysis, we will download the data and see what is the output data, what does the output data look like. So I will come back to the setting up of this pipeline in a bit. And when you set up a transcriptomics analysis pipeline, as a result, you will obtain a table of continuous numeric data that is annotated by gene ID and organized by samples in a given experiment. So your columns are going to be different sample names, and your rows are going to be different gene IDs given in the form of ensemble IDs or given in the form of transcript IDs. So the intersection will actually tell you the expression level for that particular gene ID and in that particular transcript, in that particular sample. So let's again go back to server bioinfo server and set up a pipeline for transcriptomics data analysis. This time we are going to download the data to check or to get a feel for how the transcriptomics structured data looks like and what we can do from that, or what we can learn from this data. So when you go to server.t bioinfo and under the transcriptomics section, you can click this down arrow and then click either of these projects to set up a demo pipeline. Let's click the cell line project. And here also the input files, necessary FASTQ files or input files are already been inputted, and the pipeline is already run and completed. And for the educational purposes, we have developed this demo pipeline so that you can get a feeling for setting up this transcriptomics analysis. To know more about different options or different algorithms that are involved in these different options of pre-processing, mapping onto genome or mapping onto transcriptome, and quantification of these expression levels and differential expression analysis and other annotation analysis, so you can refer to the transcriptomics, extensive transcriptomics course or program, from where you can learn a lot of information. A lot of information regarding the choice of the algorithm and how to use certain algorithms for certain data sets that we have. So let's start this process again. This is going to tell you a very basic outline of how this RNA-seq analysis is going to be performed. So it involves pre-processing, mapping, and quantification of expression level at its basic level. Yes, Omnia, I think you have raised your hand. Please share your questions in the chat. I'm following the chat window all along. So after this, you perform Trimomatic, which is going to clean, or which is going to trim out the technical and adapter sequences that are necessary for identifying our samples in the pool of samples that is being sequenced together. So let's click OK and rush through a little bit, do PCR cleaning. And then we have an option now to, for example, mapping onto genome or mapping onto transcriptome. So we will now select mapping onto transcriptome option, but you can also very well select HiSat2, which is going to map onto genome process. So BowTie2 will map the reads onto the transcriptome. And after this, we get an option, or we get a highlight for selecting the expression quantification table from RSEP. So this is going to use expectation maximization algorithm to count or to calculate or quantify the expression levels of different genes, and then give it to us in a tabular format. So let's click OK, and then we end this pipeline and click OK, and you get, yes, you get option to download demo pipeline. So from here, you'll get several different options. For example, you will get expression levels of all isoforms in the cell lines, and expression levels of expression levels of all the genes in the cell line, and expression levels of exon that are specific to exons in this specific sample. And we also get, these are all absolute quantified expression levels, and we also get a link to normalized and annotated expression level. So you can click this and download these results. I have already downloaded these, and if you are following along with me, download these results, and we will open those results up in the Excel and take a peek through what we can do with these output results. So let me open up these files in Excel. So let me share everything so that it's easy for you to follow. Okay, so I have downloaded this annotated cell line and also the expression of genes in the cell txt. So let's quickly import these files into Excel to see how the structured data looks like. Yeah, okay. Can you see my screen? I think it's still visible. Let me make it a little bit bigger so that it's not very small. So as I mentioned, yes, thank you. As I mentioned, in the on the column, you will have the sample types, and on the rows, you will have the ensemble IDs, and you find the raw counts for these expression table all along here. So if we use, or if I use, if I download the annotated, normalized and annotated table, let it open, you can quickly see the difference between these two tables. For example, this is as it has been mentioned, this is an annotated table. So what does this annotation mean? This annotation means simply the files or the samples are properly annotated. You see here, the samples are annotated by their respective names, instead of just mentioning this as the file names or the run information file names of the FASTQ files. And their values are normalized. Now we can see that their values are normalized because we don't have values like 15,000 and 16,000 anymore. Everything is between something, roughly between less than 10, and they're also negative values. So they are centered and normalized. So when will you use, for example, data in a non-normalized format and data in a normalized format? So for the downstream analysis, like statistical analysis, we can prefer to use the normalized data because most of the statistical techniques that we are going to employ will require some sort of or some form of normalization at our end. And even if we don't use the normalized data, we have to perform our own normalization